Elizabeth Schneider here, Senior Global Marketing Manager at Nanostring. We're again at the lovely Nanostring booth in the exhibit hall, 1719. And join me today as we take you on another NanoNews break. And I'm acting as your senior correspondent, coming to you live from AACR Day 2. We have an exciting day today. We're going to be visiting multiple Nanostring customers at the poster presentations to talk to them about their work using the Geomex digital spatial profiler to study cancer. Hello everyone, it's your NanoNews AACR correspondent, Elizabeth Schneider with Nanostring, Senior Global Marketing Manager. And we're here live with you for Day 2 of AACR for the morning poster session. I'm here with R&D scientist Michelle Kreiner, and she is presenting her poster entitled Spatially Resolved Detection of T-Cell Receptor Clonality Elucidates Spatial Relationships between TCR expression, immune infiltration, and cancer-associated pathways. So it sounds really interesting, Michelle. Could you first start out by telling us what is T-cell receptor clonality?
Yeah, so every T cell starts out its life with the ability to express basically an infinite array of different receptors, but during its maturation process, there is this process called somatic recombination that happens where each T cell will basically edit its genome so that by the end it only has the ability to express one single T cell receptor. And that's how our immune system gets this broad diversity of T cells that can recognize different antigens. And so your immune system starts out with this amazing diversity, but when it actually sees an antigen or a cancer cell or something that shouldn't be in your body, and the right T cell recognizes that, that T cell needs to expand to help mount an immune response. And so if you actually look at the diversity of your T cell population during this process, it will actually become a single clone, a single T cell will basically become, or a single T cell receptor will become the dominant population because that's the right one to be responding in this situation. And we're also seeing that in cancers of T cells, where the T cell actually becomes cancerous, you can actually, that original cancer cell will have its unique T cell receptor, and so you can almost use that as like a fingerprint of your cancer because all the cancer cells will express that specific T cell receptor.
Very interesting. So tell us about how can we spatially resolve T cell receptors? Like, tell us about what you guys did in the experiment.
Yeah, so for the Geomex platform, we are developing a spike-in probe panel where we make probes to the different possible T-cell receptor gene segments that are encoded in the genome, and then we try to detect them in Geomex samples to see which T-cell receptors are being expressed in a given region of interest. So this spike-in panel is designed to be used in combination with our WTA or CTA panels, so it gives the customer information. They get their transcriptomic-wide information, but in addition, they get information on the T-cell receptor expression clonality.
So you mentioned a couple acronyms like WTA and CTA, and I know here at Nanostring we love our acronyms, but if you could let everyone watching know, what do those acronyms stand for?
Yeah, so the CTA is the Cancer Transcriptome Atlas. It's a panel of about between 1,800 and 1,900 genes enriched in content or genes that are relevant for cancer and immunology. And then the whole transcriptome atlas, the WTA, covers the entire human transcriptome.
And we have that for human as well as mouse?
Yes, we have a human whole transcriptome atlas and a mouse whole transcriptome atlas. The TCR spike-in panel is specifically designed for the human, not for the mouse TCR.
Okay, so what type of samples, like what type of cancer samples or tumor biopsies were used in this study?
Yeah, so actually even before that, the first thing we wanted to do was try to validate that these probes are actually working with sensitivity and specificity that we can be confident in. And so the first thing that we did was actually test these probes on a cell pellet array where we were titrating in a T cell line into another cell line, and so we know exactly what percent of the sample is T cells, and because it's a T cell line, we know exactly which TCR segments are expressed by that cell line, and so we should be able to see, as the number of T cells goes up, you should see a signal from those expected TCRs going up as well. And so that's what we're showing in this figure. You see in different sized ROIs, as you increase the percentage of T cells in your ROI, you start to see expression of the expected, in this case it's the TR beta chain V variable region 3-1 is the one that's expressed by this cell line CCRFC-EM, and we see that there's a dose response in its expression in this experiment. So that was just to get us started to say that we're confident that we're getting specific expression and detection of our probes. And then we moved into tissue and cancer tissues. So this experiment outlined here was in melanoma. There were 3 melanoma tissues, and they wanted to just broadly profile the TCRs being expressed in these melanomas and see if they saw differences between patients or between different types of regions within the tissues.
And what were the findings?
Yeah, so they did see that there were more TCR segments detected in regions that were rich in CD3, which makes sense because CD3 is a marker of T cells. So that's what's seen here. One interesting thing is that there wasn't much of a correlation between the number of cells and the number of unique TCRs that we detected, meaning that the clonality could be different in different regions of the tumor. And overall, we generated sort of a molecular fingerprint of the TCRs being expressed in each melanoma, and we did see some similarities across patients where some of the TCRs were seen in all the patients, but then for other TCR segments, they're only detected in one patient and not the other, or only in certain regions of the tumor and not others.
So that's really interesting. It's almost like you could use the T cell receptor fingerprint to understand whether or not a particular patient is going to respond to treatment, or even looking, before and after treatment, let's say like adoptive cell therapy, like CAR T cell therapy.
Yeah, absolutely. Those are applications we're really excited to dig into.
Awesome. So if a customer wants to do Geomex and they would like to use these spike-in probes, is that an option for them?
So right now this program is in the alpha phase, so we're not offering it commercially yet because it's very much still in development. We're trying to make sure that we have all the right probes that we want in the panel and that they're performing as well as they can be. So we're not planning to do a commercial launch in 2022, but that's hopefully going to be on the roadmap for 2023. And in the meantime, interested customers can talk to us about joining our alpha program to get access to these probes earlier.
That's really interesting. I mean, as far as I know, this is really the only application of doing spatial profiling of T cell receptors within tissues. That's awesome.
Yeah.
And I thought I would add as well, for anyone who's doing bulk profiling, we do have an nCounter TCR diversity panel that allows you in a similar fashion to understand the diversity of T cell receptors present in your sample, and you can actually get a T cell receptor diversity score, where a higher score means more diversity and less clonality, and a lower score means less diversity and more clonality. So that's an option as well. Thank you so much, Michelle. It was a pleasure chatting with you about your poster. I hope you enjoy the rest of the poster session.
Thanks very much, Elizabeth.
Hello everyone, Elizabeth Schneider here again, your NanoNews correspondent, coming to you live from AACR, the morning poster session. So we're at poster number 2 of our tour of the poster session of the morning. I'm here with Cynthia Springer, Research Assistant Professor at the University of Washington School of Medicine. Today she is going to talk about her poster, Spatial Profiling of Androgen Receptor Splice Variant 7 Transcriptional Activity in Prostate Cancer Metastases. So Cynthia, I'd love for you to talk a little bit about your experiment. Why did you want to do spatial profiling of antigen receptors in prostate cancer samples?
Yes, so we worked with Nanostring to use their digital spatial profiling, and the reason we did this is because we have a nice array of tumor samples from patients that are in stage. So these samples were collected during autopsy. We have 27 patients represented and 56 metastases across those patients. And so in prostate cancer, there's a lot of difference in what is being expressed in different tumors within a patient. So not all tumors are exactly the same. And we specifically wanted to look at those differences within tumors. And so the DSP really allowed us to do that because we could select regions of interest. So within those metastases, we picked different regions of interest that have about 1,200 cells per region to look at. So this isn't single cell profiling, but it definitely is narrowed down to regions, which is great because it'll show us the diversity within the tumors. And then, so this study was initially started by Dr. Pete Nelson and Dr. Lauren Brady. And so they published that general overview of what the differences are in these tumors in Nature Communications. But then we wanted to take it a step further and look at their data and then look at how ARV7, which is a transcription factor that really seems to be upregulated in prostate cancer. So we wanted to see what its expression profile looked like in these tumors compared to the transcriptome that Dr. Nelson and Dr. Brady had already found. And so this was great because Nanostring actually had included in our RNA probes ones that were specific for AR variants. So we have ARV7, we have ARV9, and in particular when we stain, so we have an antibody that's very specific for V7, and so it differentiates it from the full-length AR. And there's a nice correlation between these DSP probes for the variants versus like the full length. So that was really nice to see that our staining was actually correlating with the increased counts for these probes. Yeah. And so then when we looked at the transcriptome in those regions that had, say, high levels of V7 staining versus low levels, what we saw was this upregulation of AR-regulated genes, which is great because it's an AR V7, so we would expect to see that. What is interesting about this, though, is that those regions that were negative for V7 still have AR full length. So that's here we did IF staining for both full length and V7 together. And so you can see that usually V7 and full length are appearing in the same cells, but sometimes there's just full length. And when that's the case, we really don't see a lot of expression of these AR-regulated genes. So it really seems like we need this V7 to then drive that regulation. And so the hypothesis is the V7 is binding to the full length, taking it into the nucleus, and increasing the transcription of these AR-regulated genes, which is then promoting the growth of the tumor cells.
I see. So is this AR splice variant 7 a potential target for therapy?
Yes, so there are companies such as ESA Pharma that are actually trying to target the V7 because V7 can't, it doesn't have a ligand binding region. So current therapies such as abiraterone and enzalutamide don't really affect V7, and so it continues to drive these AR regulated genes and drive the progression of the cancer. So the idea is then, yes, to try and target the N-terminus of the AR-V7, which would then knock out both full length and V7 and try and stop the growth of those tumors.
Excellent. So have you planned any follow-up experiments to continue to use the Geomex DSP?
Yes, so when we started this, there were only 3,000 probes, and now there's thousands and thousands.
Yeah.
Yes, exactly. So it would be great to actually go back and do this again. And especially look at this V7 with that larger transcriptome probe and see what other differences we might see between the V7 and the full length.
Excellent.
Yeah.
This is a really interesting study, and it's actually very personally relevant for me. My father had prostate cancer. He's still alive, thank goodness. My uncle had prostate cancer, and my grandfather had prostate cancer, and it did metastasize, and he passed away from it. And,
Mine
so
too.
It's something
Yes.
that's really, really relevant. So
Yes.
It's great that you're doing this work, and that the spatial profiling with Geomex DSP is enabling these
Yes,
breakthroughs.
right. It's really great. We really appreciated the collaboration between Nanostring and our group.
Excellent.
Yes.
Well, thank you so much,
Yes,
Cynthia. It was
you're
a
welcome.
pleasure, and enjoy enjoy the rest of the posters.
Thank you very much.
Yeah. Hello everyone, I'm here again at poster number 3 today of day 2 of AACR. It's another NanoNews break coming to you live from AACR with your Nanostring correspondent Elizabeth Schneider, Senior Global Marketing Manager at Nanostring. And I'm here today with Julie Gasteyer-Foster. She is the Director of Laboratory and Pathology at Global Hope at Texas Children's Hospital. And today, she is going to be telling us about her poster where they used nCounter technology to develop a custom gene fusion assay for the rapid diagnosis of pediatric cancer in Sub-Saharan Africa. I'm really excited to learn more about your work, Julie. So tell us a little bit about your assay and what was the impetus for developing this research?
Yeah, so Global Hope is a program that's based at Texas Children's, but our aim is to improve pediatric cancer care in Sub-Saharan Africa. So we have multiple treatment sites there, and we've been building up a population of doctors that can treat the kids, but we're starting to want to improve diagnosis. And so one reason that the kids don't do as well in third world countries is they're often very limited access to pathology and diagnostics. And so at our sites we have no genomics for our kids overall. Every now and then they might get a site or genetics assay, but very rarely. And so what we wanted to do was build something to start bringing genomics into the treatment of the kids. And so we looked at different platforms to try to figure out what could we realistically do on the ground there. And some of my previous work, we did a lot of gene fusion work for the Children's Oncology Group here in the US, and so it's very familiar with how frequent gene fusions are in pediatric cancer. And so we said, why don't we start there? And so we designed very large panels. We don't know kind of the makeup of the genomics of the kids in Sub-Saharan Africa. We know what's more frequent here, but we don't really know there. So we went very broadly, build a very large hematologic panel and a very large solid tumor panel, and tested it in Houston with known samples, with known fusions, about 150 samples. And we are just now getting ready, actually this week, to ship to Uganda and deploy the Nanostring nCounter as well as the fusion assays to start implementing over on site.
Oh, that's really, really great to hear. So actually, that would be, I think, the third nCounter instrument in the continent of Africa. Recently, we just had the second nCounter system, which was shipped to the Gambia with the London School of Hygiene and Tropical Medicine, and there they're actually using the nCounter to study tuberculosis. So tell us a little bit about, how the fusion assay works, and maybe the differences between doing fusion detection with nCounter versus, say, like sequencing or even FISH.
Yep. So there are pros and cons of every assay, and every time that I'm training a fellow or a student, I always want them to know you can get this with this, you can get this with this, but remember there's always limitations. And I think that one of the things we had to think hard about, again, was our resources on the ground. We don't have a big informatics team right now. We can't go straight to next gen. We don't have a lot of people trained in genomics. So we had to think about what can we realistically do. I think some advantages that why we chose this assay, we can work with very poor samples, very small amounts of samples, and with very minimal hands-on time. We don't have a large lab to do this. We have minimal folks on the ground. And so the advantage is that the poor quality RNA works well, FFPE works well. They drop all solid tumors in formalin over there. It's quick, and so within a few days we can have an answer. We probably will hope to move to next gen at some point, but we're obviously going to have to build up that capability first. The downside here is that we can only pick up known fusions, so we had to design to exact breakpoints, and we won't pick up unknowns, which we're finding here in the US all the time, but they're not the most common, right? And so we went for pretty much anything that was in the literature, including the most common and some less common, but it has to be precise breakpoints. So that's the drawback, but the way I look at it is we have nothing now. All the kids get treated the same, and we need to move towards stratifying them toward low risk, high risk, like we do here. And we can't do that without moving forward, even if it's baby steps. And so I think if you go from 0 to picking up 35 to 50% of the kids with a better diagnosis, that's a huge step for us.
Yeah, this is excellent. So I wanted to touch on something that you mentioned earlier when we were chatting before our interview, and that is that, a lot of cancer research and cancer drugs are focused towards the Western world. and you think that in the developing world, say, infectious disease is more of a problem. But actually, you were telling me earlier that, there's higher incidence of cancer in the developing world. So are we trying to understand, or do we know yet why that is, and why certain cancers are more prevalent in certain areas of the world?
Well, particularly in Sub-Saharan Africa, some of the cancers are related to viruses that are endemic to that population. And so because there are certain cancers in kids as well that are HIV related, because we went through that whole spurt of HIV in that area, those are a problem in those populations. But also their populations are very young, and so that gives you a lot more kids, just numbers of kids, and that means more cancer. So part of it is just population as well.
So are patients sort of put on different treatment regimens based on whether or not they have known fusions?
So in this country, yes. So fusions, say in the children's oncology group, if you have a child with cancer, they get a genomics workup automatically. And the different genomics play into risk groups. So are they low risk or are they high risk? If they're high risk, they're going to treat them more aggressively so that you get a better chance of cure. Whereas if you're low risk, you don't want to treat that kid as aggressively because they're going to have all the side effects of chemo as they're developing, and if you can cure them with less, let's do it, right? So certain fusions, we know in ALL, there are specific drugs that can be used to cure those kids. And in sarcomas, it's usually a diagnostic. Sometimes it actually still leads to a prediction of what they're going to respond to in terms of a drug. And that's why we want to move that way. So we work with pharmaceutical companies to get drugs in Africa, but we can't move towards some of the targeted therapies that are becoming more popular here because we don't have assays like this to even figure out which kids should get that. So they have to move together in parallel to try to improve the outcomes.
That's going to be really great. So as a final question, how familiar were you with the encounter analysis system before this? Had you used it previously in your work?
Yeah, so we did do some work early on with the children's oncology group at my previous job. We were developing similar panels, but we ended up going with NextGen because in the US, we want to get every last fusion. And we weren't
unknown fusions.
Exactly, yeah. And so we wanted all the unknowns. We didn't want to miss anything. And I think it's just a different perspective in this setting. We want to try to get the most we can for rapidly and in a sustainable way so that they could carry it out. So it's just different.
Well, I think there have been some customers that have developed encounter assays for unknown fusion. So they essentially develop custom probes upstream and downstream, but you need to know potentially where the purported junction site could potentially be. So I don't know if you've thought about using that at all.
Yeah, so we're actually doing some additional studies, kind of the next phase 2. So we're implementing the fusions in Uganda now, but in Houston we're working on some additional assays to try to keep working at the genomics of pediatric cancer. We're looking at copy number changes. We're looking at point mutations. Again, only things that'll either be prognostic or potentially targetable later on. We're also looking at some subclassifications for lymphomas, which are very big in Sub-Saharan Africa. And some of those may be differences. They use 5' versus 3' probes of the same gene to look at overexpression. You can predict a fusion that way as well. So.
Yeah, exactly. Well, thank you so much, Julie. This is really, really exciting.
Thank you.
It's great to see this technology being used to have an impact, where there otherwise, like you said, wasn't any option for these children.
Yep, we're excited about the possibilities. Thank you.
Excellent. Thank you so much, and enjoy the rest of the poster session.
Thank you.
Hello everyone, and we're back with poster number 4 today of our tour of the morning poster session, day 2, coming to you live from AACR in New Orleans, Louisiana. I'm your NanoNews Break host, Elizabeth Schneider at Nanostring Technologies, Senior Global Marketing Manager. I'm here today with Corinne Ramos from Biotech, where she's the Director of Research and Development. And she's here to talk about her poster, Spatial Biomarkers to Improve the Predictive Power of Response to Immunotherapy in Operable Lung Cancer. So Corinne, I understand that you use both nCounter and the Geomex Digital Spatial Profiler in this study.
This is exactly what I do. Actually, I just wanted to have, like an unbiased way of looking at the gene regulation. So that's why, like we use the encounter first to get, like an idea about, like in the bulk, like what will be the upregulation of gene. And after that, we use the Geomex DSP platform, which is, like the digital spatial profiling platform. I don't need to tell you this. To look, like at more predictivity in terms of treatment response within, a very specific region of interest, like on the tissue. And we have used non-small cell lung cancer tissue, where we correlate the transcriptome activity, the special transcriptome activity to the response actually to the therapy. And that was the PD-1 therapy that those patients received.
Was that pembrolizumab?
Exactly, that was an anti-PD-1 therapy.
I see. And so what did you find in your results?
So we found, like in terms of the transcriptomic, we found, that the signaling of T-cell exhaustion was present in the progression disease patients, so the patient that did not respond to the therapy.
That makes sense.
That makes sense. And for the one that completely responds to the therapy, we have seen in terms of the protein content, in the spatial location, in the context of the disease, we have seen, like infiltration of T cell, and as well, like activation of those T cell, and the presence, like of the drug target.
Oh wow! So it seems like in those patients who responded, the immune cells were able to infiltrate the tumor.
Absolutely, and this is exactly what you are seeing here when you do see on that heat map an upregulation of all the proteins that are really involved in the T cell infiltration and activation.
Great. So what panel did you use for the encounter analysis?
For the encounter analysis, I don't even remember one panel.
I think it's
We have used the tumor signaling 360 panel.
I see.
So
did you get the tumor inflammation signature scores as well?
Yes, and this is something that I'm not showing here, but we had the tumor infiltration score actually within the patient that responded to the therapy.
So I'm guessing the patient who responded probably had a higher test score?
Absolutely, absolutely.
Excellent.
Yeah, we're not showing this actually in this paper, but we have looked, like at this very specific signature, which is included, like in that panel.
I see. And with the Geomex experiment, did you segment out the tumor versus the tumor microenvironment?
This is exactly what we have done, like in this one, we have used the automatic segmentation algorithm from Nanostring, from the Geomex, where we could, automatically segment, like the tumor compartment versus the stroma compartment by using fluorescent antibody, the PanCK in that case, and the CD45 for the stroma compartment. So that's what we have done here, but sometimes, like we go a little bit further and we try, like to segment only, like a very specific cell population.
I see. So have you thought about going beyond protein and doing like the whole transcriptome?
Yes, so this is next, and this is, like my next poster, which is not, like for today, but obviously, like we have a lot of requests in my biotech for the WTA, which is the unbiased, like transcriptomic assay, just because people, they just want, like to browse, like completely on the tissue section, on the different, like region of interest, what could be, like in a completely unbiased way, the signature in terms, like of transcription.
Yeah, it's really interesting. So we do actually have, when you mentioned T-cell exhaustion, on the Encounter platform, we do have an immune exhaustion panel.
Yes.
And on Geomex and Encounter, actually, you can profile T-cell receptor diversity.
Absolutely.
So we can do that spatially. It's actually a special spike-in, custom probes you can spike into the Cancer Transcriptome Atlas for RNA or the WTA. So that might be something that you'd like to do in the future. We have it as an alpha program right now, where you can work with us to get those T-cell receptor probes.
I'm very interested, actually.
Yeah, actually, and if you get a chance to get away, actually, I think in section 27, we have a poster by a nanostringer, Michelle Kreiner, where she did exactly that. She looked at the prevalence of the different T-cell receptor sequences within the tissue section using Geomex, spatially looking at.
Okay, so that's something, like I definitely want to do, because I think, like they will still different, like in terms of phenotype within those T cells, and you might want actually, like for looking at potential, like new target, drug target, you might want to look, like at the TCR on this one. Yeah, that's great, super.
So can you tell me a little bit about what you guys do at EMA Biotech?
So EMA Biotech, we are an imaging company, so we're very much tissue-based. So everything, like which is imaging and especially, like related, we are providing the services at pharmaceutical company. So we have, like imaging mass spectrometry technology, the Geomex DSP platform, obviously. We do have single cell technology as well in terms of proteomic profiling. We have the Hyperion system from Freedyne. And yes, so our specialty, it's really, like the spatial bioanalysis and spatial biology. So basically we can look, like at the accumulation or distribution of the drug, like on the dissection, and with this platform go there and profile, like in terms of transcriptomic, proteomic content, try to understand, like this mechanism, response of the treatment, prediction of the treatment, everything, like spatially related.
Great, and it sounds like you might also be interested in the cosmics, spatial molecular
And
imagery.
yes, I am.
Yeah, it would be a great addition.
It will be a super great addition because we know that going actually towards, like single cell in terms of transcriptomic, while keeping, like the context of the while keeping the spatial context, it's really the must. And we're very happy actually, like to be collaborating with you on this one as well.
Excellent. Well, thank you so much.
Sure,
Corinne.
no problem.
It was a pleasure.
It was
chatting
a pleasure.
With you,
Thank you
and
very much.
Enjoy the rest of the poster section.
Thank you, thank you, thank you very much. Thank you.
Hello, everyone. I'm back again. Elizabeth Schneider, Senior Global Marketing Manager at Nanostring, reporting live from AACR for another NanoNews break. Today, I'm with Ajit Johnson-Nirmal, who's a postdoc in Peter Sorger's lab at Harvard Medical School, and he's here today to talk to us about the spatial landscape of melanoma progression at single cell resolution. So this is really exciting, Ajit, because I know spatial biology is on everyone's minds right now. So can you walk us through what you guys did for this experiment?
Sure. So what we're trying to do here is to build a pre-cancer melanoma atlas, and this is part of the Human Tumor Atlas Network. And what we're really trying to understand here is the early spatial and molecular events that allow melanoma cells to proliferate and grow into a full-blown tumor. And in this case, what we have done is to combine multiplex imaging data, a method that was developed at Sorger lab called SciSIF, and then we combined that with microregional transcriptomics data, that includes Geomex, and also high-resolution 3D imaging data to understand or build a map of what's happening in this early precursor lesions to query them to understand the underlying biology of how cancers proliferate from normal skin to a more invasive lesion. So I'll just, as I said, this is more of an atlas work, so I've kind of highlighted a few vignettes that I thought were interesting in this poster here. So to start with, we initially built a computational algorithm which uses something called a latent dirichlet allocation modeling, a method to identify recurrent cellular neighborhoods or communities. And what I mean by communities here, it's essentially trying to divide your tissue into regions of structural or functional importance. So in this case, once you identify those communities, you can query them very rapidly across multiple patients or across multiple tissue types. And in this case, what we are showing is that we find these communities become increasingly complex as you go from normal skin to a more invasive state. And a very interesting finding here is that we find evidence of immune suppression as early as the precursor stage. And this is really important because this pushes our understanding or our evolution, our need for looking for progression-related risk signatures further back into evolutionary time. And what was also interesting here is that when you look at the normal and precursor or these early lesions, you don't necessarily see a difference in the cell types that are present between these regions or the actual abundance of cell types. What we do see is the interaction pattern between these cell types change from normal to precursor. So you really need the spatial resolution to be able to tease out on these signatures as tumors move from normal to a very early precursor stage.
Interesting. So this would suggest that you should do screening, in this case for melanoma, even earlier potentially to pick up on those immune suppressive signatures.
Yes.
And to potentially be able to treat it at an earlier stage.
Yeah, so from a clinical perspective, a surgeon would essentially shave this off and potentially that will never recur. But from a biological perspective, to understand what is causing these lesions to eventually progress into an invasive tumor, these kind of analysis are very, very important.
Yeah. And for the Geomex data, did you do whole transcriptome?
No, this was using the CTA. We regularly do
WTA now.
WTA now, but this was I think 2-3 years back when we had access to the early access to the CTA program.
Yeah, yeah. And tell us a little bit more about the 3D imaging. Like, what technology do you use for that?
So in this case, we have used a delta vision microscope, which does optical sectioning and then computational deconvolution to reconstruct into 3D. Again, these are just 5 micron section. So we use, I'll probably get into on why we do this. But currently what we are doing is we are trying to increase the thickness to 20 microns or 30 microns to capture a few more layers of cells. As from this initial analysis, we have seen that a cell can have multiple interactions, and it's not necessarily a one-to-one interaction when a cell like macrophages, especially within the tumor microenvironment.
Yeah. So I wanted to ask you, you mentioned that as the tissue progresses from normal to precursor to cancerous, that the interaction between the cells changes. But how are you really studying that interaction, or how do you know that that interaction is changing? Is it just the spatial location, or are you looking at like secretory proteins that are, secreted? How do you measure that?
So it's a twofold explanation. So obviously with the spatial data, we can infer whether a particular cell type is interacting with another cell type across the whole image, and then we can really narrow down on regions where we find these interactions. And if we want to understand the biology, what we do is obviously that's when Geomex comes into play. We actually identify those ROIs and do a whole transcriptome analysis or TCA and identify what are the pathways that are different in these regions which are not in other regions where they do not interact.
I see, I understand. And so one final question, you talk about single cell resolution. You're obviously aware of the cosmic spatial molecular imager that's coming out later this year. Are you guys doing any experiments with that?
Yeah, so we are very interested in cosmics, and obviously the 100-plex protein assay and the 1000-plex RNA sequencing assay. I think there is some collaboration between Peter Sauger's lab and Nanostring, and hopefully we'll be able to work together in the future. And I'm really looking forward to use that much data to get better understanding of these early precursor lesions.
Yeah, I mean, I think it's really important to catch it as early as possible, right? Yeah. So, thank you so much, Ajay. It was so nice to talk to you.
No problem.
And best of luck with your work, and we're excited to see what comes out of your lab.
Thank you.
Hello, everyone. It's Elizabeth Schneider coming to you live from AACR Day 2 for another NanoNews break. I'm your Senior Global Marketing Manager at Nanostring. Today we're here for the afternoon poster session with Shyla Bonnet. She's an R&D scientist at Nanostring, and today she's going to be telling us about a really cool new workflow called Spatial Proteogenomics, where you can detect RNA and protein on a single FFP slide. Now this is super cool. I mean, everyone either usually analyzes RNA or protein. It never really does both, but there's so many different things that can occur, post-translationally. That proteins can get degraded, they can get modified, they can have like phosphate groups added. So I'm really curious to see how this is actually done. So, and this is spatial profiling as well in tissue. So can you walk me through like how we went about, trying to come up with this assay?
Yeah, that's, thank you, Elizabeth, for, your interest. So as Elizabeth alluded to earlier, this assay will allow you to detect and quantify both RNA and protein from the same slide. You get away from a lot of those technical variations that may be occurring, but also allows you to understand the interplay between the two analytes. So we did a three-pronged approach for this assay. The first one is focusing mostly on the technical development, because we need to know whether or not if these two analytes are going to play nice together, because they both want two different conditions.
Exactly.
So we had to find that middle ground, which was a balancing act, but doable. And so this is the first part of it, of the poster. Kind of goes over that technical development. We looked at staining order and how the impact of formamide that is involved with the stringent washes for RNA has a negative impact on protein. So based from that information, we decided to move forward with ISH before IHC staining approach. The second issue at hand was proteinase K. It's ideal, or it's required for optimum RNA detection, but it's a protease, so it's going to chew up your protein target. And so we needed to kind of find that happy medium. What was going to make the RNA happy and what wasn't going to kind of make the protein upset. So that's what we did here. We just kind of titrated up protein, and as expected, high concentrations of proteinase K is detrimental for protein, while it's making RNA quite happy. So we found that we decided to go with 0.1 microgram per mil of proteinase K because we maintain as much as possible the protein performance while minimizing the impact on RNA detection. So based off of all this information, and there were other things that we did look at, and we do have a white paper in which those other technical development assays are included. But in the end, we came up with a simple workflow, and we tried to make it as simple as our original assays, We did have to add an extra step because you're now having to deal with two different types of probe sets. So day one, your normal DWAX and epitope retrieval.
And this is with the whole transcriptome atlas or cancer transcriptome atlas.
That is a very good question. Yeah, you could do this with WTA, human or mouse, and then we also have CTA. So we just tested this on mouse content, and it's working. All this information shown here is dealing with FFPE. We tested on fresh frozen. So, we're trying to make it as compatible for many different sample types. So then day two, your normal-ish, workup. Then we start to transition into protein. And then from there is pretty much your normal workflow.
And then this is readout on an Illumina next-gen sequencer,
Yes,
right? Okay.
yes. And so because we're using, at least with respect to the human, we have a 147-plex protein content.
Okay.
And using NGS, it allows us to have higher plexing capability.
Got it.
Also, it allows us to distinguish RNA and protein based off their barcode. So,
Got it.
So which was critical.
So did we try first a cell pellet array and then we tried FFP?
Yeah, and so we use cell pellet array as our go-to sample to evaluate the performance with respect to sensitivity and specificity, and we benchmarked it against the respective single analyte control.
Got it.
And so we just kind of looked at, we compared our, cell line, target for both protein and RNA, but like what a lot of the scientific realm is interested in, how are they correlating? Even though correlation, even noted in literature, can be poor between the two analytes, but there are some targets that do correlate well, and that's what we'll see here with CD45. And I should also say, we also compared the two different approaches. The multimodal, in which you extract data for both analytes from the single slide, and then your multi-omic, where you integrate two separate data packages to see how it compares. And so, which is critical, because it's like, why would multimodal be better than multi-omic? So as you can see, the multimodal, we do see is comparable to the multi-omic. But we do get correlation between a number of targets, and we do, and this is supported in literature, we get positive correlation for CD45 between the two analytes and negative correlation for fibronectin, and that is consistent with literature, so which is great. But in the real world, we don't use cell pellet. We're all about tissue. So we tested on various types of tissues from disease to normal. And so we just provided a single example for non-small cell lung cancer.
Okay.
And so again, we're just looking at sensitivity is comparable. We do get a little loss, but a lot of that loss, as reflected here in this histogram, are those targets that are low expressors that are right on our threshold. So it's like it's still there. It's just your whatever you set your threshold at, and we're very stringent. So.
And it looks like we did segmentation between the
That's up there.
and the tumor.
Yeah, so we did that with colorectal cancer. So this is where we masked and collect for our tumor, which is green. Then we went ahead and masked and collected for our immune. And so this plot actually allows us to look at the differential expression of targets within the two segments for both RNA, which is the cyan, and protein, which is the pinkish type color. And then again, dealing with the or like evaluating the correlation. And then here, because it's beyond correlate, how do analytes correlate, but how do other targets track with one another, what's going on. And so these two heat plots allow us to see how certain protein targets, which are listed on the y-axis, track with RNA targets. So here is like a prime example. We have one RNA target that's involved in mucus production, correlating with autophagy type targets, which are part of mucus, that correlate with mucus production. And then we have anti-correlation with tumor targets in which, they should not be correlating. So it's very consistent with literature.
This is really exciting. I mean, I feel like a lot of researchers are going to be really interested in this.
Yeah.
It cuts down on time, right? It saves time to get more information.
Yeah, and it cuts down on the technical variation that occurs between serial sections, because you're capturing the same cell population, which could differ between the two sections. So,
and I think another thing to point out is because Geomex DSP uses light to profile and section different areas, it's not like laser capture and microdissection. Your sample is not destroyed.
Exactly.
So you can still save it and image it, and use it for another analysis. So that's really great. Well, this was really great, Shyla. Thank you so much.
Thank you.
And as you mentioned, there is a white paper on this workflow, and it is available on our website, so you can have a look. And have an enjoyable rest of the conference and
Thank you.
poster session.
Okay, thank you.
Hello, everyone. I'm back, your NanoNews correspondent, Elizabeth Schneider with Nanostring, Senior Global Marketing Manager, coming to you live from day 2 of AACR at the afternoon poster session. So today we have Asia Legrini. She is a graduate student at the University of Glasgow, and she is presenting on high-plex proteomic prognostic marker discovery for patients with pancreatic ductal adenocarcinoma using digital spatial profiling. So, Asia, tell us a little bit about, what was sort of the driver behind this project? What were you trying to understand better?
Yeah, okay, so pancreatic cancer is a really deadly cancer. There's very little biomarkers used in the clinic, apart from BRCA. So really what we were trying to look at is developing new predictive and prognostic biomarkers for this disease.
And why did you choose spatial profiling? I understand you use the Geomex. So what aspect of the Geomex digital spatial profiling platform did you really like for this study?
So what really attracted us to this was the spatial aspect. Obviously, pancreatic cancer is a really heterogeneous disease, and so we really wanted to sample multiple different regions to see if there is a spatial relationship between different histopathological regions within the disease. So that's what really attracted us to the Geomex.
I see. And so were you able to profile the tumor and the tumor microenvironment and segment out each region of interest?
Yeah, definitely. So we use a pan-CK positive mask for the tumor areas and then a pan-CK negative mask for the tumor microenvironment areas, to really elucidate the differences between the tumor and the tumor microenvironment, which we're really interested in.
And were you doing protein or RNA with the Geomex?
Okay, so for this particular example, we're doing protein, but we are doing RNA. So we've done a whole range of all the different panels, including the whole transcriptome atlas.
Oh, great! You used the whole transcriptome atlas. Excellent. And so, what did you end up finding when you did the experiment?
Yeah, so we were really particularly interested in the immune population. So we did the immune oncology panel for this particular example. And so we found that there was an immune high population and an immune low patient population. And we found that there was significant differences between the prognosis between those two populations. And we also found a B7H3 marker, which is really interesting. So we're looking to explore that further in a future patient.
And what types of cells or what pathway is B7H3 associated with?
So it's an immune checkpoint marker, so that's why we're really interested in it. There's a lot of buzz about potential immune checkpoint blockade.
Excellent. So do you have any follow-up experiments that you're going to do with the Geomex? It sounds like you have been using it for other projects.
Yeah, so we do a lot of the RNA panel work actually at the moment. So we've just done a whole transcriptome atlas, and we're doing that between treatment naive and neoadjuvantly treated patients.
Excellent. Well, thank you so much, Asya. It was a pleasure. I hope you enjoy the rest of your poster session. Thank you so much.
Thank you.
Hello, everyone. I am here at our last poster session of the afternoon here at day 2 at AACR in New Orleans, Louisiana. I am your NanoNews correspondent, Elizabeth Schneider, Senior Global Marketing Manager at Nanostring. And I'm here with Ana Oliveira, who is the Director of the Spatial Biology Core Facility, as well as the Molecular, Immunologic, and Translational Sciences Core at University of Virginia. She's also an Assistant Professor in the Department of Pathology. And she's here to present her poster on spatial transcriptomic evidence for an inflammatory pro-tumorigenic microenvironment in normal human dense breast tissue. So this is really interesting. So a lot of the posters here and studies are looking at cancer tissue. So why were you interested in looking at normal dense breast tissue?
Well, I think it starts, every scientific community needed to look for the pre-cancer tissue, the pre-cancer condition.
Yeah.
Because if you prevent the cancer, you have a more, how can I say, it's better for the patient if you have prevention than you needed to treat the cancer.
It's better to prevent the cancer than to try and treat it once it's there.
You needed to start more studies about it, using the normal tissue.
You know, that's a really great concept. I mean, I think we focus so much on the treatment. We're really trying to understand, well, what does normal breast tissue look like?
Yeah.
How does it function? So that you can understand the moment that things start to go awry and you can try and treat it there rather than waiting until you have a full-blown tumor that's developed.
Exactly. How is the moment that you is normal and then you move for not normal breast?
Correct.
When it's happening.
Yeah.
Why some women develop cancer and other ones no?
Yeah.
Why do you have difference of me? That's I think you need to know now, and you need to study more. You need to look for the normal. What's the normal? No one knows what's the normal. You know exactly what is when you have some disease. I think it's
so.
Good.
How did you decide what areas of the tissue to profile?
Yeah, here I use on the normal breast, but this women has a dense breast. It's not some condition that some women has.
Yeah.
The dense breast is risk factor for developing cancer breast.
Okay.
And then if you look here, you can see, for example, here is the fat tissue, is a non-dense breast. And here you see the stroma more dense, that you don't have a lot of the more fat cells.
I see.
And then is the dense breast. And then you select the dense breast, dense tissue, non-dense tissue, and the interface between them.
I see.
To analyze.
I see.
And this is a very nice platform because first you can stain your tissue, use 4 morphological markers to understand what kind of tissue you have, what you want to select. For example, if I want to select immune cells, I use a specific marker for that. And here you use vimentin and SMA and
cytokeratin.
Yes. And then you could select on these different regions to analyze and compare.
Now, did you do any additional sort of segmentation between, say, the vimectin portions versus pancytokeratin when you did your analysis to look at the transcriptomic expression in the vimectin areas versus pancytokeratin, or did you just look at the whole area?
I used the whole area. I not to do the segmentation analysis.
Okay, I see. And tell us, you were looking at RNA.
Yes.
And what type of assay do you use on Geomex? Was it whole transcriptome atlas, the CTA?
Yeah, in this study I use the CTA. That's a panel that have 1,800 targets.
Yep.
There are many targets specific for cancer and then specific for immune pathways that I want to analyze. What's the how can I say the cells is behavior here? What's the immune cells behavior in this both situation, and what difference between them? I think in this panel ask my questions.
Yeah. And what sort of differences did you see between the dense and non-dense breast tissue?
Yeah, I think the more important difference is the immune cells. In the dense breast, you can identify more targets involved in suppress immune pathways.
I see.
And when you look at the non-dense, you see more inflammatory targets.
I see.
So what the conclusion is, you have more. You have around the dense, you have non-dense and more inflammatory process. Maybe try to resolve the situation, or no, maybe this non-dense becomes dense in the future because have the chronic inflammation. That's we need to study a little more.
Interesting.
But it's very interesting use that it's possible today to see that in the microenvironment you have more microenvironment in the different
and so
profile.
Do you think it's important or possible to somehow reverse dense breast tissue to become non-dense?
I think no. I think
no.
Revert no, but maybe you can stop the process.
I see.
If you identify some process or some proteins or transcripts that are involved in the
transition
for breast cancer, maybe you can stop.
Yeah.
I think you have here two possibilities. One, to find the targets to show you, look, this dense breast maybe becomes a cancer. Then you have the transition for cancer. And then maybe you can find some targets that say to, look, you can treat this before it becomes a cancer. And you have two possibilities.
Yeah.
The treatment, pre-cancer, and to find the signal that's, pay attention, you have something different here. You need to be careful.
Yeah. It's really interesting work. So are you going to do any follow-up studies with the Geomex DSP?
In the future?
Yeah.
Yes, I think I want to use the new equipment to have more, to validate some targets.
I see. Mm-hmm.
And I want to increase more donors, more samples to have the more strong
database.
Yes.
Excellent. Well, thank you so much, Anna.
Thank you.
It was a pleasure.
Pleasure
I hope
for
you
me.
enjoy the rest of the meeting and
too.
enjoy the poster session.
Sure. Thank you so much. Nice to meet you.
Hello, everyone. I'm Elizabeth Schneider, Senior Global Marketing Manager here at Nanostring, coming to you live from AACR, another NanoNews break. So, we saved the best for last. I thought that our last poster was the last poster presentation of the day, but we managed to track down the infamous Arutha Kulasinghe, Senior Research Fellow from the University of Queensland. He's come all the way from Australia to present his poster on the multi-omic dissection of immunotherapy response groups in non-small cell lung cancer. This is actually a collaboration that we did with Nanostring. So Arutha, tell us a little bit about your poster. So why did you choose a spatial biology platform to analyze these non-small cell lung cancer samples?
Yeah, great question. One of the challenges in spatial biology, well, in immunotherapy at the moment, is there's a lack of predictive biomarkers. So we thought that spatial could potentially help in that space. We had a really great cohort of 90 lung cancer patients retrospectively collected, 45 which were given immunotherapy and 45 standard of care. And so we had really good survival data for these patients, so which patients responded and those that didn't respond to immunotherapy. And we were able to apply spatial to those to sort of delineate signatures associated with response and resistance to immunotherapy.
Excellent. So I know that you use the Geomex digital spatial profiler. So what was your selection strategy for profiling different regions, and did you segment within each region?
Yeah, great question. So the strategy we used was, because it was a tissue microarray, we used tumor stroma. And the way we did that was we gated on cytokeratin and non-cytokeratin. And when you do that, you effectively capture the tumor and the stroma from every TMA core. So that's what you can see here. We used cytokeratin to capture the tumor regions and CD45 and non-cytokeratin to capture the stroma.
Okay, excellent. And then what assay were you doing with Geomex?
Yeah.
Was it CTA, cancer transcriptome atlas?
Yeah, so we used the CTA and the protein of serial sections.
Oh, great.
Yeah,
so you're probably really excited about the spatial proteogenomics workflow
yeah,
where you can do it on the same
exactly,
section.
Yeah, no, it was incredible when I saw that because we've been wanting to get that same readout of a single section, and we're really excited by that assay.
Oh, great. So what did you find?
Yeah, great question. When we looked at the tumor regions, so the cytokeratin positive regions, we could identify proteins here that were enriched for between the response and the non-response groups, and the ones that were significant are the ones in the blue. So this is looking at the protein level, and this is at the transcript level. What was really interesting for us was when we looked at the stroma, so the non-tumor regions between the response and the non-response groups in the RNA compartment, so the mRNA, we could see that there were two really standout markers. So IL-2,
I can see that.
yeah, and SPP1. So SPP1 is osteopontin, and when we developed a multivariate signature for that, which is you're using multiple markers here to look at that signature, we could see that IL-2 by itself discriminated that cohort. So that meant that IL-2 stromally located separated out your responders from your non-responders, right? So, which was amazing to see in this test data set. And the ROC curve has a 0.9 AUC. So that was, it's potentially showing you the power of spatial for biomarker discovery. And we're doing a lot of work now to validate these findings.
Yeah, because if you looked at bulk analysis, you may have missed that,
yeah,
right?
Exactly.
It would have gotten hidden.
Yeah,
so
I mean,
you essentially found like a needle in a haystack.
Yeah, exactly. Yeah. And because we can compartmentalize the signal to tumor or stroma, it's a broad way to really, discover biomarkers using the strategy. So yeah, we're very excited by it.
And then what about osteopontin? Like, it's been a while since I took biochemistry, but osteo is in the name.
Yeah.
So it has something to do with bone growth?
Yeah, so it plays a number of roles, and I won't jump into what it does exactly, but it's got multiple functions. So we're trying to now really hone in and understand what is that underlying biology and why is that in the stroma, and in the stroma together with IL-2. So IL-2 is known to drive in your CD8 cells into the tumor microenvironment. So we think that is what's promoting the response group. So IL-2, when found in the stroma, associates with response to immunotherapy, at least in this cohort.
I see. And so with the responders, though, did you see the IL-2 in the tumor regions, suggesting no, you did not?
No, it was in the stroma exclusively.
Interesting. And did you also find signs of immunosuppression in the non-responders?
Yeah. So we're working through those at the moment. Yeah.
Okay, great. And so you use the CTA for this. Since then, have you used the whole transcriptome?
We have. I haven't shown the data here, but we've got whole transcriptome data on the back end of this as well.
Excellent. This is really great. So thank you so much, Arutha.
Absolutely.
And I also just wanted to say that if you haven't gotten enough of Arutha, you can come to the Spotlight Theater tomorrow morning at 10 a.m. in Theater A. And I hear you're presenting on some COVID-19 stuff.
Yeah, at a cancer meeting.
Yeah!
It's going to be fun.
Can you give us a little teaser of what we can expect?
Absolutely. So we've got some fascinating data with the Cosmics, where we've used Cosmics to subcellularly annotate COVID-19 lung tissues, and some fascinating findings on the cell types that are being infected and the immediate neighborhoods within that.
It's really interesting. Well, I mean, I think that, COVID-19 research has really opened up this world of immunotherapy in infectious disease.
Yeah.
And some of those drugs that are used for autoimmunity or used for checkpoint inhibitors are being repurposed,
exactly.
For infectious disease.
Yeah.
So I think it's really cool that, this resurgence in infectious disease research due to the situation of the pandemic is really having a halo effect on other areas of research.
Yeah. And I think the conversation is also there for COVID and cancer,
Yes.
and COVID and lung cancer, for an example, right? You're getting a lot of lung cancer patients who are getting COVID. What does that mean? So I think it's a blending of those two disciplines, and it's an exciting time to be doing work in both those fields.
Exactly, and with COVID-19, you have a lot of tissue damage.
Yeah.
You have a lot of restructuring of the tissue.
Yeah.
You have fibrosis that's occurring, and that occurs in cancer as well.
Yeah.
So there's a lot of crossover.
Absolutely. Yeah. So a lot to learn. Yeah.
Excellent. Well, thank you so much,
Thank you.
Aretha. It was my pleasure. Thank you for coming all the way from Australia to
No,
this
no problem.
AACR meeting.
Thank you.
Thank you so much.